All right, guys, so I got a few emails about some confusion for regulation of gene expression. So I've put together a quick video to go over some of the main points that are relevant to the AP exam, might be relevant to any exams you're going to take. I can tell you that free response questions really like to ask about gene expression. So even if you're not going to be completely tested over it in class, you should definitely know the basics and how gene regulation works in eukaryotic and prokaryotic organisms. As always, here's the what you must know. So these are the main points about gene expression that we're going to be talking about. If there's any embedded videos, make sure that you watch them. I can't go through them on here, but you should definitely check out the class website. Look at the videos that are there. AP Bio Khan Academy has some really good videos related to gene expression to check out to help you out. So let's talk about the regulation of metabolic pathways. So when I talk about metabolic pathways, we talked about cell respiration and we talked about photosynthesis. Those are two metabolic pathways. And if you remember, we talked about how funny it was if you would have to memorize all of the enzymes that are involved, but each step in those processes requires some sort of protein or some sort of enzyme. So when we're talking about gene expression, we're talking about how we're regulating pathways in your body. So if we take a look at this diagram right here, sorry, there was a hair on my iPad. We see a lot of words and we see a lot of arrows, not errors. So I'm going to start with this word up here, precursor. Now, precursor is a starting point. So a precursor for a protein would be an amino acid. A precursor for a carbohydrate would be a monosaccharide. So those are just things that are little building blocks that are going to turn into a bigger product. Now, cells vary the amount of enzymes that they need by regulating gene transcription. So if you remember, DNA has to turn into mRNA, and then it will turn into a protein through a series of steps. The DNA to RNA step we call transcription, and the mRNA to protein step we call translation. So your cell is going to regulate expression and regulate enzymes by regulating the transcription part, the actual DNA to mRNA. That has to leave and go to the ribosome and make a protein. We have genes that can be turned on and can be turned off. So we have on genes and we have off genes. Now, I would want to turn on a gene because let's say I need a particular enzyme or protein to be made. I want to turn that gene on that's responsible for making it. But let's say that I have an abundance of a certain protein. It doesn't make sense for my body to make more. So I'm going to turn off that gene. And when I turn that gene off, transcription isn't going to happen and translation won't happen. So I'm not going to make anything. I already have what I need. I don't need to make more. That's what we call the off gene. And on genes, I want something to be made. I need transcription to happen to get that protein because that's the first step. Those would be my on genes. So if you look, you can see this precursor is going to be turned into a final product through a series of enzyme reactions. Now, let's talk about operons. So an operon is a cluster of genes that are related to each other, and they contain an on and an off switch. So when we talk about pathway enzymes, pathways are all related to each other. So when I talk about the cell respiration, that is a pathway, and all of those steps are dependent on each other. So there is a series of switches that are occurring. So when I talk about operon, the cluster of related genes with on and off switches, and these would be all the enzymes for a particular pathway. All enzymes in a pathway. An operon contains three parts: a promoter, an operator, and a gene. These all constitute an operon. So let's start with what a promoter does. Promoters are where RNA polymerase attaches. And if you remember, RNA polymerase is responsible for starting the transcription. So it takes that DNA, it's going to read it, it's going to turn it into RNA. That RNA is eventually going to head off to the ribosome for the translation step. But right now, all we care about is transcription starting, the first step. So a promoter is where the RNA polymerase attaches to the gene. So in my diagram, I have the promoter. This whole region is the promoter. It is where RNA polymerase is going to attach itself, this little teardrop looking piece. So the promoter is where RNA is going to attach itself to the gene to start transcription. So I'll make a little second legend here. A promoter is where RNA polymerase is going to bind. It's going to be the spot that transcription starts at. That's the transcription start point. Now the second piece is called the operator. So I'm going to use a different color. That's kind of the same color. Here is the operator. Now you can see everything is kind of divided into chunks. Here's our second piece. The operator is where the on and off switch would be, and it controls where RNA polymerase is allowed to go. So RNA polymerase is going this way. The operator dictates how far it's allowed to go, how much gene it's going to have access to, and how much it's allowed to transcribe. So the operator is our on/off switch, and it's going to control access. Our final piece in this operon, again, I'm talking about all the pieces that make up an operon. I'm just talking about the pieces of an operon, the 3 pieces. The last piece is the gene itself. Genes code for enzymes. Remember, a gene is a segment of DNA. That DNA codes for some sort of amino acid, and when they're put together with other amino acids, we make a protein. So that gene is going to code for something. I need to transcribe it and translate it in order to make my final product. It could be a protein, it could be an enzyme, it could be a ligand, it can be anything. So my final piece are the actual genes. Those genes turn into proteins. They code for them. And you can even see in the picture, gene A made amino acid A. Gene B made amino acid B. Gene C made amino acid C. We put them together, we make a polypeptide, which is another name for a protein. So we just talked about the 3 parts that make up the operon. Now let's talk about the different types of genes that can be experienced. We have regulatory genes. Regulatory genes produce repressor proteins. So when I say I'm going to repress something, I'm going to stop it, or I'm going to lessen the thing that it does. Repressor, you can say stop, you can say slow down. So when I say I'm going to repress the amount of transcription that a gene is going to experience, I'm going to lessen the amount it's going to do. So repressor proteins, these genes, regulatory genes, they're going to produce a repressor protein. Remember, genes turn into proteins. They are not the same thing. A gene is a segment of DNA that codes for the protein. So we're going to produce a repressor protein that's going to bind to the operator to block RNA polymerase. So RNA polymerase is just having a great time. It's headed down the operon, and then it comes into contact with the operator. When it does that, it's going to be blocked. That repressor blocks it. It's going to block RNA polymerase. It can't keep reading that gene. It's going to fall off. Now, we have two types of operons that we deal with. We have a repressible operon and we have an inducible operon. So we just talked about what the word repress means. So a repressible operon is going to take a gene that is on and it's going to turn it off. We are going to stop transcription. And when I stop transcription, I stop translation too. I'm not making anything anymore with a repressible operon. So we go from on to off. And we're going to talk about the specific examples related to them in detail. But in this particular picture, you can see the promoter region. That's where the polymerase is going to bind. You can see the operator, which dictates how far it's allowed to go. And you see the genes that need to be transcribed. When I have a repressible operon, a molecule called a co-repressor. So it's going to, co means work together, it means it works with it. It's going to bind to that operator. RNA polymerase can't reach the genes. It can't read them. It can't transcribe them. It's going to fall off. It won't make anything. So one particular example that we're going to talk about, and you're going to see a lot for the rest of your life if you're going to go into biology or anything medical, we're going to talk about tryptophan. So when I talk about a tryptophan operon or the TRP operon, this is a repressible operon. This is one that the gene is turned off. I'm not making anything. I have inducible operons. Induce, so if you think of like pregnant people, when they're induced, we're forcing them to have the baby at that point. So we're going to take an off gene and we're going to turn it on. So this gene was originally off. It wasn't making anything. I'm going to turn it back on. It's going to start to make stuff. So here is my operator again. Here's the promoter where RNA polymerase is allowed to attach. Right now, it's off. Whoa, what the heck? Right now, it's off. There is something there blocking it. But this is inducible. So that piece is going to fall off, open up that operator, and we can continue doing our transcription. So when we talk about an inducible operon, we're going to talk particularly about the lac operon. And that lac stands for lactose. All right, back to our operons. So we're going to continue working with the repressible one. Remember, this is a gene that is usually turned on, but we're going to turn it off for some sort of reason. We're going to turn this off. You usually see this with anabolic situations. Remember, anabolic means to build organic molecules, and the opposite, catabolic, tear down. That's when we break them down. So we're going to build a bigger product when we talk about anabolic. So in a repressible operon, we're going to add that co-repressor onto the operator to block RNA polymerase from working. We're going to turn that entire operon off. We're going to stop what it's doing. We're not using it anymore. When we talk about a particular example of a repressible operon, our favorite one to use is the TRP operon. TRP stands for tryptophan. So TRP and tryptophan go together. So let's kind of look at it a little bit more in detail. So again, our three pieces that make up the operon, three related pieces to each other. Got the promoter, that's where RNA polymerase is going to bind. You've got the operator, that's going to dictate how far RNA polymerase is allowed to go on this gene. And our third piece are the actual genes itself. So let's take a look at this entire setup that we got going on here. So I've got DNA. I've got the genes. Remember, genes are segments of DNA that code for things. And I see I've got TRPE, TRPD, TRPC, TRPB, TRPA. These are all individual genes that code for an individual amino acid. And when those individual amino acids come together, it's going to make a protein. Okay. Now, when tryptophan is absent, versus when tryptophan is present, the operon does different things. So let's talk about, my body has a lot of this substance, tryptophan. What happens if I already have tryptophan present? Do I need to make more of it? Do I need to make more of those individual pieces to make this tryptophan? The answer is no. I don't need to make more. So in the TRP operon, when tryptophan is present, this operon is going to turn that entire thing off. We're going to shut down. We are not going to make, we do not need to make the enzymes necessary for building tryptophan anymore. Oh, ran out of room. When tryptophan is present, we will switch into that off mode. We will turn into a repressible operon, and I'm not making the individual pieces anymore because I already have the substance present. I don't need more of it. So in the tryptophan operon, we are going to turn off. Tryptophan operon repressible turns off. Now let's look at that. Here is that operator blocking the RNA polymerase. We are not going to make RNA. We're not going to translate anything. We have blocked it. It's off. TRP operon is off. We went from on to off. We are not making any more. Now the opposite is the inducible operon. It's usually off, and now I'm going to turn it on. This is a catabolic process. Remember, that's to break down. So one of the things that we talk about with inducible operons is we talk about lactose. Lactose, a sugar, ends in O, so it's a carbohydrate. We're going to turn this operon on in a certain situation. So what happens if lactose is present? Well, lactose is a big sugar. I need to break it down into its individual monosaccharides in order to digest. So I need to turn this operon on to make the pieces necessary to break down that sugar. So if I have lactose present, lactose is present, then I need to make lactose digesting enzymes. I'm going to remove that part that attached to the operator, that repressor. I'm going to take it off. That's going to allow my RNA polymerase to get moving, and it's going to start to make these genes transcribe to their individual amino acids: beta-galactosidase, permease, trans-acetylase. These are enzymes that are going to break down lactose. So, real quick, lactose is present. I need to build the enzymes necessary for breaking it down. I'm going to turn that whole operon on so that I can start getting to work making those enzymes that are going to be responsible for breaking down that lactose. The opposite was true in that tryptophan. I had tryptophan already present. I didn't need my body to make more of it, so we're going to turn that entire operon off. So repressible, turns off. Inducible, we're going to turn on. Two different situations, two different results. Now let's talk about negative and positive controls. So when we talk about negative controls, the operons are switched off by some sort of a repressor protein. You saw those little Lego pieces attaching to that operator to turn them off. When we talk about positive controls, these are proteins that are going to interact directly with our genome to increase transcription. And I have two examples here. I've got CAMP and I've got CAP. So one example in particular is low glucose, low blood glucose. OS again, ends in OS. It's a sugar. We should remember that. CAMP, a molecule we're going to see in a minute, is going to bind to a CAMP receptor protein. And remember, we don't like writing things out all the time, so we're just going to call it CRP. And it's going to activate transcription. So let's get a little bit more into this CAMP and this CAP and this whole positive control notion. So CAMP accumulates when glucose is scarce. So there is not a whole lot of glucose in my body. It's going to accumulate. By accumulate, I mean it's going to build up. CAMP binds to CAP, catabolic activator protein. When I activate CAP, it's going to bind to DNA upstream of the promoter. So we talked about the difference between upstream and downstream. So here's a stream. Here's you. Smile. When I talk about upstream, here is upstream. It's before. When I talk about downstream, it's after you. Here's downstream. It's after. So CAP is going to bind to DNA. So we'll now relabel our thing. You are now DNA. CAP is going to bind upstream or before that promoter. It's going to increase the affinity of RNA polymerase to that promoter. When we talk about affinity is not the same thing as infinity. Affinity means the tendency to stick with or be around. So I have an affinity for being around my dog. I like to be around my dog. I have an affinity for being in bed by 10 o'clock. I like to be there. Affinity means attach, interact with. So we're going to bind to DNA upstream of that promoter. We're going to increase the affinity of RNA polymerase to the promoter. So how much it wants to be together, going to make it really want to be together. As a result, I'm going to increase transcription. I'm going to start making RNA from that DNA. Increase transcription. When I increase transcription, guess what else is going to increase? The amount of RNA I'm going to make that's then going to be able to be translated into amino acids to make a gene. So if you are slightly confused, slow down, go back to everything I just said, click on some videos, start watching some stuff to help you understand. All right, so we've talked about how a lot of your DNA doesn't really code for anything. It's just junk DNA. It's left over. It's conserved from evolutionary changes. When we look at your typical human cell, only 20% of your genes are expressed at any given time. So there's 80% that are turned off and they're not doing anything. Now, different cell types are going to turn on different genes to carry out specific jobs. So my liver cell has a different job than a brain cell. Different genes are turned on to allow that to happen. When we talk about those differences in the two cell types, we talk about differential gene expression. Differential means different, differential. So each gene has a specific function, and every cell has specific genes that are going to be expressed with that function. So again, DNA, specific functions are turned on for that specific cell type. Here's a nice picture of how our DNA is organized, and we kind of talked about that with Unit 4. So we're getting more into some review, some bridging the gap between Unit 4, when we learned about DNA, to now. And we're going to talk about the structure of DNA. So you know that DNA is a double helix. And inside of your body, we need to organize that DNA into more compound little pieces. And remember, we talked about package them tightly together and you make some chromosomes. Tightly packed DNA. Now, depending on how tightly packed together your DNA is, will dictate how correctly or how strongly your genes are going to be expressed. So when we talk about chromatin structure, when DNA is tightly packed down together, it's harder for it to be transcribed and then translated because it's hard to get RNA polymerase into those spots to read that DNA. So tightly bound DNA is less accessible for transcription to occur. So we need to do some stuff to that DNA to unwind it. and one of those is called DNA methylation. So we're going to add methyl groups to DNA, and that's going to cause them to be tightly packed together, and we're going to lower the transcription. So methylation is going to make it hard for me to transcribe because everything is so packed together. And RNA polymerase needs to get into those little spots and spaces and read. So in order to get that DNA to loosen, I have to do histone acetylation. So if you remember, so I'm going to kind of look at the picture at the same time. You have histones, little circles, that this blue, maybe I should use the correct color, this blue DNA is wrapped around. It helps them organize. I need to unwind DNA so that I can reach those individual pieces to read them. So acetyl groups are going to be added to histones, and that's going to cause those histones to loosen up DNA, loosen the gaps, and increase transcription. So I need to get inside to touch that blue DNA. So here, all of that DNA is tightly wound. Nothing can reach that DNA to transcribe it. I need to reach it. So we're going to go through histone acetylation. We're going to unwind and allow those transcription factors to reach the DNA and get the transcription process going.